everyone. Thanks for coming. It's my distinct pleasure to introduce our speaker, Dr. Giacomo Bernardi. He's a professor at UC Santa Cruz. I'll keep it brief since we're a little bit behind schedule, but a little bit about Giacomo's background. He did his master's degree and his PhD at the University of Paris, where he was studying genomics and population genetics of fish. He then went and did a postdoc at the Pasteur Institute, correct? That's where right. You also said that was part of your mandatory military service. That's right. So I don't know what he was doing. Maybe he's designing some genetically modified killer fish for national security interests. Probably something like that, right?
I could tell you without that.
Yeah. And after that, Giacomo came to Central California. He did a postdoc at Hopkins Marine Station with Denny Powers and decided this was an area that he really liked. And after that, started the position at UC Santa Cruz, where he's been for 20 years. His main background is looking at evolution, population genetics, ecology of fishes, in pretty much every ocean in the world. He works in the Mediterranean, California, Polynesia, Micronesia, Madagascar, Mexico, almost anywhere you could think he's been there, collecting fish, and doing a lot of interesting work. Today he's going to talk to us about some work he's been doing looking at the genetics of some invasions by some species called the Lessepsian fishes. And if you don't know what that is, Giacomo is going to tell you.
Yeah. Thank you so much. It's great to be here. Thank you so much. It's great to see all those friends. And I thought that since I've taught here a few times in the past 20 years about mostly California fishes and also maybe Polynesian ones, I thought that I had not yet talked about another things that I work on, which is biological invasions in the Mediterranean. So for those of you who have heard my talks before, I thought that I would try to diversify a little bit and pick that topic. By the way, before I forget, the idea of fish tacos is great. I very much like that. So, I'm going to try to rush because I know that you want to get out by 5 and my talk is super long, so I'll try to talk quickly. All the pictures, by the way, that I show are of tropical fish, but they are all taken in the Mediterranean. So, this is the outline of what I will be talking about today. Brief history of the Mediterranean fishes, tropicalization of the Mediterranean in the last few years, Lessepsian invasions. Most of you probably have never heard of this before, so I will explain what it is. The genetics of Lessepsian bioinvasions, some details about all the different things that we do, and future plans and prospects. A brief history of Mediterranean fishes. So this is the Mediterranean. It is a relatively small sea that is surrounded by a lot of different countries, which politically makes it very complicated. It is the most impacted and the most polluted and the most bioinvaded sea in the world. So that makes it, in a sense, interesting, but also a little bit daunting. It has not always been like this. Right now, you're familiar with the hotspot of biodiversity in the world. That is the Coral Triangle between Indonesia, Papua New Guinea, and the Philippines. But over the history of our oceans, while the center of biodiversity is currently in the Coral Triangle, it has actually migrated over the millions of years. And about 40 million years ago, it was actually centered around the Mediterranean. There is evidence of that, fossil evidence of that, especially in a very famous Eocene deposit that is close to Verona in northern Italy, in Monte Volca. And in that place, you see a lot of very nicely preserved fossils that look just like coral reef fishes that we're very familiar with, just as further evidence of what I was just talking about. Now, what happens is that the Mediterranean at that time was the northern end of the Tethys Sea, and the southern end was closed, and then the Strait of Gibraltar was closed with Africa collapsing into Spain, and little by little, the entire Mediterranean dried up completely. So there were no fish left in the Mediterranean. That's what is called the Messinian Salinity Crisis, MSC. And about 5 to 6 million years ago, the Strait of Gibraltar opened up again. There was apparently this huge waterfall that lasted probably 1 to 200,000 years and filled back up the Mediterranean. Because of that, the Mediterranean Sea is a relatively recent body of water and relatively depauperate, considering that it's a relatively recent body of water. There are about 500 or so fish species, only 50 endemics, about 10% of them are endemic. So because of that, it's a place that is rather vulnerable to biological invasions, maybe also because a number of ecological niches are still relatively open because there are relatively few species that have invaded them, let's say. And just as an example of that, in the Mediterranean, in the native Mediterranean, there are only 2 herbivorous species. One is a parrotfish that is found mostly in the eastern and southern basin, and the other one is a bream, Sarpa salpa. So this gives you an example. If you go to any coral reef where you see tons of herbivores, this is not at all what you would see in the Mediterranean, where there's only 2 and really one is most common, the bream. Now, because of global climate change and the warming trend of the globe, there is what is called the tropicalization of the Mediterranean. The Mediterranean is getting warmer, and this is the regular state of the Mediterranean, where the eastern basin of the Mediterranean tends to be warm, but with global warming, very often we have episodes where the entire Mediterranean gets very, very hot. And depending on the forecast, the amount of time it will take for the entire Mediterranean to be very warm is forecasted at different times, but essentially it's where we're going into that is a very warm entire Mediterranean. So that's what we have to contend with at this stage. This has 3 main consequences at the level of fish species, which is what I tend to focus on. The first one is what is called the meridionalization of the Mediterranean, which means that southern species tend to move northward. So there are northward invasions of southern species that used to be in the warmer part of the Mediterranean, tend to be found now as far north, say, as the southern coast of France, for example, in the case of, for example, Thalassoma parvo. There are some Atlantic invasions, some warm-adapted species. This arrow doesn't really do justice to what's really happening. It's mostly species that are along the coast of Africa that are warm-adapted, that tend to enter the Mediterranean and invade from this side. Not very many species, but a dozen species or so. And then what we're going to talk about today is those species that are entering the Mediterranean through the Suez Canal. These are called the Lessepsian invasions. So today there is a lot of super interesting things to talk about in the Atlantic and the northward invasions, and I wrote a few papers about this, but today we're just going to focus about one of them, which is probably the more critical part of the biological invasions of the Mediterranean as far as fish are concerned. I don't know if John is here, but you are probably familiar with the whole story of ballast water and stuff. This is something that is really important for invertebrates, but for fishes, it's not as important because fish larvae in general don't do all too well in ballast water. And so because of that, the invasions that are found in the Mediterranean by fish are usually not due to ballast water. Okay, so now we are going to explain a little bit more about what Lessepsian invasions are. So this is the northern part of the Red Sea. There are 2 gulfs. This is the Gulf of Aqaba. This is the Dead Sea. This is the Gulf of Suez. The town of Suez is right here. This is the delta of the Nile River, and this is the Nile. One very important, a disclaimer right away, one really important piece of the puzzle of what I'm going to talk about today is a piece that is very poorly known, that is what is happening in the Gulf of Suez. Working in the Gulf of Suez is nearly impossible for military reasons. I work with a lot of Israeli colleagues, and I work with a lot of colleagues from Arab countries, and I cannot mention anything to each other because for political reasons. And when you are working around here, it's dangerous. You know, I mean, people have Kalashnikovs all over the place, and it's just dangerous. So sampling here is not great. The great advantage, though, is that permit requirement is not exactly a big issue. So, you know, you go there, you grab fish, you bail out. That is not the issue. This is really not. But anyways, so what is really interesting about this story is that the idea of building a canal is really old. So all pharaohs apparently built some canals with some deviations with the Nile River. Then Napoleon tried to do that as well, but failed. And it took a long time for the Suez Canal to be effectively built. So the opening was in 1869. The engineer was Ferdinand Lesseps. And so because of that, one of the first scholars of Lessepsian invasions, Francis Poor, who died about 2 weeks ago, named the fish that move from the Red Sea into the Mediterranean Lessepsian migrants in honor of the engineer of the canal. The opening of the canal itself, I don't have time to go into this, but it's fascinating. And understanding the history of the opening of the canal is interesting if you want to have any understanding of the current situation of what is going on in the relationship between the West and the Arab world right now, actually. I don't think it's an overstatement to say that. Aida, the opera, was written for the opening of the Suez Canal, for example, and there's a great book that is called Parting the Desert that talks about the intrigues around the opening of the Suez Canal. What is interesting about this place, Ferdinand Lesseps here, this is his statue. He was born in Versailles, which is my hometown, and when I was a kid, this statue was right outside my house. And so I always wondered, I say, who is this guy? And I didn't think that one day I would end up studying quite a bit about him. But what is interesting about the situation is that the Red Sea has about 3,000 species of fish. There is about 500 in the Mediterranean. And the salinity is higher in the Red Sea, and the temperature is higher. The delta of the Nile is right here, and the Nile outflows here and then wraps around and goes this way. So the front of the opening of the Suez Canal, which is here, is almost completely fresh water. And so because of that, when they opened the canal, there were very few possibilities for fish to cross from the Red Sea into the Mediterranean. There are a few species that go the other way, maybe one or two at the most. So they are called anti-Lessepsians, and there are so few of them that people don't even mention them. But there are one or two. There is a truckload that goes the other way. I'll talk about that in a second. So here there are some bitter lakes, which have high pH. Here there's a lot of fresh water, and all this is a way of preventing any sort of natural invasion of organisms from the Red Sea into the Mediterranean. Things changed when, in the late 60s, Nasser decided to create the Aswan Dam that would regulate the flood of the Nile River. Without going into the details, it was a bit of an ecological disaster, and also it was not an economical, it was also an economical failure. But what the main result of that is that a lot less fresh water started flowing out of the Nile, and the natural freshwater barrier to migration stopped there. And a lot of organisms, including fish, really started to invade the Mediterranean in earnest after the creation of the Aswan Dam. And this is true also for invertebrates and for algae, as well as one or two seagrasses. So things really change a lot. Currently, because of the warming of the Mediterranean, the last thing happened, that is the ocean became warmer and became a lot more similar to the Red Sea. And so currently, here and here, the places are much, much more similar than when the canal was opened a little over 100 years ago, 150 years ago. So the result of that, the net result of that, is that there is currently more than 300 species of marine organisms that have gone from the Red Sea into the Mediterranean. And last time I counted was about 90 species. By today, there could be 100. This is 20% of the native species of the Mediterranean are now from the Red Sea. So that is a very, very large impact. In general, when you're talking about biological invasions, there are a number of predictions and expectations that have been proposed. Usually, successful biological invaders are invading open ecological niches. And in the Mediterranean, there is a number of them because of the natural paucity of organisms. There is low competition. Think about tons of herbivores, for example, from the Red Sea that are going into a place where there's only 2 species. There is low predation because they don't have their natural predators. And in general, there is low parasitic load. There are also genetic expectations, which is what I'm interested in. The genetic diversity is supposedly related to success. So when you think about the number of invaders that come in, if there are few individuals that are invading, theoretically, their genetic diversity compared to the original population is relatively low, and potentially these would not be very good invaders. Instead, when the genetic diversity is high, then they may be able to invade the place. This is very difficult to test in our case because there is a number of invaders that probably go into the Mediterranean that go unnoticed. And the ones that are successful that we actually can see have already gone through this first filter. So it's very difficult to know if maybe actually there is 500 species that go in there and only 1 or 2 make it, or if they all make it. It's very, very difficult to test that. And the main reason for it is because we cannot work in the canal zone. So this is counteracted by bottlenecks, which means when very few individuals are going into the Mediterranean, you are expecting low genetic diversity in the Mediterranean. And this, when there is a bottleneck, the only way to counteract bottleneck is when the population grows very, very fast. For genetic reasons, this has to do with the genetic breakdown of breeding of the invading species. Population growth needs to be very, very high. So that's one of the predictions. And another thing that may counteract bottleneck is if you have very strong selection. So if the individuals are not doing very well, but there is very, very strong selection, you can select just the genes that are very, very effective at making the species be a very good competitor, and then they will be successful. So we're going to test all these various things and see if any of these pan out or not. So now we're going to really talk about the genetics of Lessepsian bioinvasions. So there are 90 species of fish that have invaded the Mediterranean, but very, very few of them have been studied, and they have not been studied very thoroughly. I know that because I did most of them. And in the case of other organisms, it's even less so. I mean, there is like I said algae, but it's not even an alga, but anyway. There are inverts, I mean, there are very, very few species. So there's a lot of room for studies. You know, there's, as in other areas, but this is definitely in need of more work. This is just a picture to remind me that it's really interesting that the species that have invaded the Mediterranean are from a lot of different families. It's not like one family, for example, of fish would do very well and you would have 20 species of that family that would go in the Mediterranean. Not at all, it's just the opposite. It's almost like if you were cherry picking like one species here and there, and then you see all these guys, they're all in different families, and it's kind of in itself very, very interesting. So I'm going to talk just about a number of things in the genetics. I'm going to talk about species identification, cryptic species, population bottlenecks, a little bit about genomic approaches. So first, species identification. So anything that is found in any other field applies to biological invasions, with the difference that in biological invasions we know even less than for regular species, because we have in general few individuals to look at. This is one example of them. So these are, they are called sweepers. The genus is Pempheris. They are a bit of a nightmare. I have to say that I don't like them at all. And I've spent like the last 2 years studying them, and I hate their guts because they all look exactly the same. 90% of them in GenBank are misidentified, and 98% of them are misidentified in museums. So they all look the same. So the one in the Mediterranean was supposed to be a species called Pempheris vanicolensis. And these are the two vanicolenses that are found in GenBank. This is the one that is found in the Mediterranean. It's not at all vanicolensis, and it's another species called rhomboidea. So it's misidentified. This seems trivial, but in fact it is quite fundamental, because if you don't even know what you're looking at, it's going to be really difficult to do anything about it. And there is a lot of management issues. There have been ecological disasters that have been brought by Lessepsian fishes. Just to give two examples. One is just last week, an entire family in Turkey died because they were eating pufferfish from that they saw this big fish. They said, "Well, this is great." They all ate it. They all died because it came from the Red Sea and they were not familiar with it. And another thing is that you have herbivores that are rabbit fishes that entered the eastern basin. They have completely replaced the local herbivores, and they have mowed the entire region, and so the urchin population went down. There is no more urchins, and then themselves collapsed, so the fisheries are completely collapsed in that area. So, I mean, they're quite consequential. This is a DNA barcoding study that we did on Lessepsians. Lessepsians are not very easy to find. There are 90 species. Here we collected 43 of them in Lebanon, and barcoded them, which means that we sequenced the cytochrome oxidase 1, which is the barcoding gene that is used in a lot of different species, and tried to determine at least if the identifications were correct. What we found is that 44% of the names that were used were incorrect, or at least different than what was expected, and 28%, these guys, have a genetic distance to the one that they are supposed to be that is more than 2%, which means that it's a different species altogether. So 44% are misidentified, and 28%, we don't even know exactly which species they are. They're probably a different species altogether. So we have 90 species of fish, and we're not exactly even sure of what they are. That's essentially the bottom line of this slide. Cryptic species, in general, in the sea, there is a number of species that can be identified genetically, but when you look at them morphologically, they look exactly the same. This was originally seen with invertebrates by Nancy Knowlton, and she suggested that there were a lot of cryptic species in the sea, and she was very right, and it was quite a foresight. And in the case of fishes, it's the same thing, where you would have, you collect a bunch of fish, they're all the same, you sequence them, and then you realize that there are two species, and you cannot quite figure out why there are two species, but more importantly, you don't know if they are ecologically different and if it's ecologically relevant. And the reason why you can't do that is because they look the same. So when you're looking at them, you don't even know which species you're looking at since they look the same. So you could have ecological observations of 1,000 individuals, then kill them all, sequence them, and then go back to your observations and figure out what each species is doing. This has never been done for reasons that I can imagine. So that is not great. However, in our case, we found something that was kind of interesting. So this was done with the blotch-feet dragonet, which is a Callionymus, a small species. And this is a situation where there are actually two cryptic species. So the way these fishes were collected was really interesting. These were collected with a beach seine. So we went out with some students, put a beach seine out, seined, had a bunch of fish, all in the same seine. They all look exactly the same. Sequenced them. They are in two different groups. There are two different species. We sequenced a bunch of genes. There are two different species. So they live exactly in the same place. They live in sandy habitat. It's really difficult to do much ecology with those things. So that's the way these guys were collected. And what is interesting about it, though, is that the individuals that are from the Mediterranean are all in only one of those two species. So the blue guys are the individuals that were collected in the Mediterranean. The red guys were collected in the Red Sea. So after we seined and we sequenced the Red Sea ones, we had those two groups, those two clades. But when we sequenced the Mediterranean ones, we only found them in one of them. So there has to be something different about those two species because apparently only one of them was able to go through the Suez Canal and not the other one. And we don't know what it is. So we kept all the voucher specimens of every single individual, and I've spent days at the microscope looking at every single one of those guys and trying to figure out what was different because I was convinced that something would be different and we had missed it. And I could not find anything. I could not find any differences whatsoever. So I don't know, something must be different, but I don't know what it is. Or this is just with another gene, and that's the same deal. Okay, now for population bottlenecks. What is predicted is that when you have a situation, it's a great experiment. I mean, if I had dreamed of an experiment, I could not have dreamed of a better one. You know, I'm going to build a canal, put a bunch of fish from one place to another, and then look at them. This is great. So what is done is that you have these fish that are going from the Red Sea into the Mediterranean. And what you're expecting is that few of them will go through the canal, and these guys will be a genetic subset of the original population. That is the definition of a bottleneck. Actually, not exactly. The definition of a bottleneck is what happens later. At the beginning, you have some individuals from the original population that are going to another place. At the very beginning, these guys carry the same genetic material as the original population. So far, nothing has happened. But as they start interbreeding, there is inbreeding going on, and this is where the genetic breakdown happens, and this is when the genetic bottleneck happens. So you need to look at it after a few generations to be completely precise. Another thing that I need to mention is that the way these organisms go from the Red Sea into the Mediterranean is unclear. For the longest time, I thought that larvae would go through the canal. But at least in some species, this is not the case. These are adult migrations. And the reason, the evidence for it, is that some parasites have been found in Lessepsian migrants, that is Red Sea fishes in the Mediterranean. And those parasites are Red Sea parasites that can only be carried by adults. They cannot be carried by larvae. So the only way for those parasites to go from the Red Sea into the Mediterranean is to hitch a ride on an adult. So at least for some species, adults do migrate. So population bottlenecks, is there any evidence of bottleneck? I thought, well, this is kind of trivial. Of course there would be evidence of bottleneck. But the reality of it is that there isn't. Very early on, there was a study that was done using allozymes, lowly allozymes, but this is just as good as anything else. And in this study, Upenaeus is a species of goatfish. They did not find any evidence of population bottlenecks. So people thought, well, those guys are not using markers that are refined enough to see bottlenecks, and so obviously they're not going to see them. But this has been the pattern for most species, not the exception. This is an example of a silverside that is called the hardy-head silverside. And these are the Red Sea species, the Red Sea individuals. These are the Mediterranean individuals. If there was a bottleneck, you would expect all the Mediterranean individuals to be all grouped into one genotype. And instead here, it is just as diverse as the original population. There is nothing special about it. And this, incidentally, is an invasion that occurred a very, very long time ago. Athyrinomoros invaded, I don't know, more than 60 years ago, if I remember correctly. This is another example of my favorite, the sweeper. The Red Sea individuals are the red genotypes here. The blue, light blue, and dark blue are the ones in the Mediterranean. And as you can see, they are spread all over the place. So you don't even need to look at the details. Here's just to show that there is no evidence of bottleneck, really. They are just as diverse as the original population. So it has not been found in all these species, which is essentially all the species that have been studied. There is no bottleneck. So that's the pattern that we see, is that there is no bottleneck. So this may be due to three main reasons. One is that maybe what happens is that we're looking at old invasions. And so that's why actually we cannot see a signature of bottleneck, because this happened a very long time ago. Another possibility is that there is constantly multiple invasions. So first you have a bottleneck, but then there is other guys that show up and they are bringing more genetic material. Later on there are other invasions, and it happens so often that at the end you don't have any signature of bottleneck. And lastly is that instead you have invasions of very large numbers. So testing this is not very easy, but we have enough cases to actually look at all these different situations. So that's what I'm going to show in the next few pictures. One is Scarus goban. Scarus goban is called the blue-chinned parrotfish. If you've been taking the class in the Sea of Cortez, you have seen this species. It's a species that is found everywhere from the Sea of Cortez all the way to the Mediterranean now. But it's a species that invaded the Mediterranean very, very recently, just a few years ago, about 5 or 6 years ago. And because of that, there are very, very few individuals. So this is to test if it's an old invasion. So this is from Lebanon. We were able to get 5 samples, and I think in Lebanon only 7 individuals have ever been seen. So it's really, we are doing our best to get as much as we can. And all 5 of them had 5 different haplotypes. So it's not because you have an old invasion that you're going to have many haplotypes. This one is very recent. These guys arrived just a few years ago, and they are just as diverse as the ones that are found in the Indian Ocean or in the Red Sea. Another possibility is the idea that there are multiple invasions. So this is difficult to test, but we tested it with one of the rabbit fishes, Siganus luridus, which is called the dusky spinefoot. It's a very good fish to eat, but it's also, when you get stung, it hurts quite a bit. It's found everywhere now. So it entered the Mediterranean in 1964. There's a sister species, Rivulatus, that entered before. This one entered later in 64. And okay, it's spread all over the eastern basin. It's found everywhere here, and that's the one that is the main culprit for having removed all the algae in that area. But what happened in 2003 is that on the island of Linosa in Italy, a number of individuals showed up. Linosa is an island where fish surveys are done very extensively. And the reason why they are done extensively is because one of the main scientists that works on biological invasions has a side project happens to have a house in Linosa and surveys fish there all the time and knows all the fishermen. So in 2003, he heard that there was a major recruitment event of Cyganus luridus that just showed up. And we know that's exactly when it happened. So this was to determine if multiple invasions are the reason why we cannot detect bottlenecks. So there was one recruitment event that showed up at that time. And so what we did is that he shot them all. I mean, he went there, shot them all. It was like no prisoners. And here they are. They are found all over the place, and there were not any less diverse than their original population. So I understand that they may have come from a lot of sources from the Eastern Basin, but in one recruitment bout, there was just no evidence of a bottleneck either. So what we looked at finally was the blue-spotted cornet fish. The blue-spotted cornet fish is kind of interesting because it's a fish that looks nothing like anything else in the Mediterranean, so it's really conspicuous. So it showed up in the Mediterranean in 2000. We almost know exactly when it showed up. It's very conspicuous, and in 5 years, invaded the entire Mediterranean. So it was a really interesting species, and this one showed some evidence of bottleneck at the mitochondrial level. It ended up into 2 clades. At that time, we only had 1 marker, and we saw just 2 haplotypes. Since then, a number of sequences have been done by other groups, and they found that they fall into those 2 clades. There are 2 clades of cornet fish, but they essentially fall into here and here, and that it might be some mild evidence of bottleneck. So we thought, oh, this is a great species. We're going to start working on that guy. So the historical distribution is very well known because they are so conspicuous. Every time one shows up somewhere, everybody talks about it because they look so different than anything else in the Mediterranean. So it was really easy to figure out exactly what happened. So their spread happened rather quickly. So over the years, these are all the records over the years, and they ended up invading the entire Mediterranean. There is no other Lessepsian species that is everywhere in the Mediterranean. This is the one species that in 5 years filled up the entire place. And we also have a pretty good idea of exactly how it happens. There is a southern route that was used, and then a northern route that was used, and then also a middle route maybe. And so with these precise records, we can exactly have a history of how the invasion occurred. So everybody said it's a sprinter, including myself. It's a sprinter, it's amazing, and it does some super cool things, and this is very unusual. And then I thought about it a little bit more, and I said, is the spread of Fistularia commersana truly unusual? So I thought so, but then last year, Michel Gaither, who studies biological invasions of grouper and snapper in Hawaii, published a very interesting paper where what she did is that she was able to predict the rate of invasion according to the native range of the species. And this is how it works. So all these points each point is one species, one invading species. And on the y-axis, you have the continuous extent of spread, which is what is the distance of spread of the species. So in our case, it would be the entire Mediterranean. So this is like thousands of kilometers, right? And on this side is the FST. FST is a measure of population structure in the native range. In other terms, this means that when the species has a very low FST, which means that the species is called panmictic and can disperse very well in its natural range, then you are predicting and actually observing that it has spread a lot as a biological invader. Instead, when there is a lot of population structure, which means that the dispersal of the species in its native range is not very big, so for whatever reason, maybe it has very short larval life or other reasons, then in fact, the prediction and the observed thing is that the extent of the range will be very, very small. So what we did is that we collected individuals of the cornet fish all over its range, which means in close to La Paz in Mexico, and then Hawaii, French Polynesia, Seychelles, and in its native range in the Red Sea. And we could use genetics of the native range to determine what the population structure of this species is in its native range, and then plot back this value on the original plot and say, okay, now we have the natural FST, we know the extent of the particular extent of the spread, where is it? And those three dots here correspond to the northern, the southern, and the middle route in the Mediterranean. And in fact, it is within the 95% envelope of this correlation. So in fact, as spectacular as the spread of that species is, it is not outside of the prediction by this correlation, not at all. So that was really interesting. Finally, I'm going to talk about genomic approaches. So I don't have much time because we had to choose between eating tacos or talking. And I think that the choice was a good one. But I'm going to try to still talk about these things because it's pretty cool. So we used rad sequencing. I'm not going to go into the details. If you know what rad sequencing is, you don't need to know about it. And if you don't know what rad sequencing is, I'm just going to say 2 words about it. The idea is that you are sequencing very small portions of the genome randomly placed all along the genome. So instead of having 1 or 2 genes that you're looking at, you are looking at tens of thousands of small sequences that are found all over the genome. That's essentially what we're doing. It's just to have a really good representation of the genome, the nuclear genome of the species. We used 44 individuals from the Red Sea, 31 in the Mediterranean, and we use what is called an 8 cutter. So the enzyme that is used has 8 nucleotides. This means that on average it cuts every 65,000 base pairs. So the genome of Fistularia is 1.2 gigs, which you're expecting about 30,000 sequences, 36,000 sequences. So we found 31,200, so it's 32,000 sequences. So that's roughly, the expectation was great. That's exactly what we had. So first of all, we wanted to know if there is evidence of bottleneck, but now not looking at just one gene, but looking at 30,000 of them. And so the number of usable loci is slightly different in the 2 things. I'm going to go straight to the important thing. The heterozygosity, which is a measure of genetic diversity of the population, is 0.13 in the Red Sea, 0.14 in the Mediterranean. Without knowing anything about these data, all I want to say is that these 2 numbers are essentially the same. This is really interesting because once again, it shows that the invading individuals are not any less diverse than the original population in the Red Sea. The theta, which is the genetic diversity, is also very similar. And the FST, which is a fixation index between the 2 populations, is extremely small, which is what you are expecting in a highly dispersive and low population structure species, which Fistularia is. A structure plot shows the same thing, which is the Red Sea and the Mediterranean individuals are the same. If they were genetically different, one would be red, the other one would be green. And instead, the fact that everything is red here means that there is no population structure between the 2. There is no evidence of genetic bottleneck. Now, one of the things that I was puzzled about, I was wondering if maybe what we were finding was that there is a family of individuals that is getting together in the Gulf of Suez and just going across, and all these individuals would be related. So we can actually test relatedness among the individuals that are found in the Mediterranean. And this 4.6% of the individuals in the Red Sea that we sampled had some degree of relatedness, and 3.7 in the Mediterranean. So I thought that I was going to be super smart and find some great stuff, and once again it failed miserably. They were not more related than expected. So that was another thing that was not all that great. So last but not least, one of the things that might be the key to success to those species is that there is strong natural selection on the individuals that are going to be the successful invaders. And it's possible to test that when you have wide genomic sequences as we do. So the way you do it is that you calculate the average FST of each. You calculate FST for each of those loci, which is we have 30,000 of them or so. And then you do an average. And for theoretical reasons, you can actually identify the ones that are under selection because they are called outliers, which is that they are completely on the side of the distribution curve. Because when there is selection, the fixation index of those loci is higher than expected. This is the distribution of the FSTs. It's very close to 0 as I've shown before. This arrow here is where the level of 3 standard deviations is. And so anything that is to the right of that is supposed to be subjected to directional selection. And in this case, it's 200 loci. There are 200 sequences, very short sequences that I have, that are potential candidates for selection, which is not many, but actually plenty to look at, which is great. So I want to show you what the population structure used to look like with all the loci. Actually, when you look at just those 200 loci, by definition, there is strong population structure. And so this is what you are finding with just looking at 200 loci that are under selection. So there's a strong fixation in the Mediterranean compared to the Red Sea. Now, you take those 200 loci and you're going to blast them against GenBank. There is a number of fish genomes that have been sequenced, and so you can compare those sequences with what has been sequenced and determine if it corresponds to anything that is interesting. So 66% were recognized as fish sequences. 29% were recognized as fish protein coding genes. And this is really interesting in itself because the expected, if there was nothing else going on, is 4%. So in those 200 loci, you have an excess of protein coding genes compared to what you are expecting. And what is interesting is that 8% of them are actually specifically involved in osmoregulation. So that is not 8% of 29, 8% of the 200. So osmoregulation seems to be like a very important factor. I say, oh wow, this is really great, this is very interesting. And there's a lot of other things to talk about right now that I just don't have exactly time to talk about, and it's very, very interesting. So I talked to some colleagues of mine, and there is a paper that now is in press by Valeriano Paravicino and Yoni Bellmaker, who are modelers, and they modeled what is the fate of Lessepsian biological invaders in the Mediterranean according to different scenarios of global climate change. They looked for temperature, acidity, and salinity. And what they found is that the strongest predictor of success in expansion of a spread in biological invaders was salinity. The main reason for fish not to be able to invade the Mediterranean is actually a salinity barrier more than anything else. And so when I mentioned that we had genes about osmoregulation, they almost fainted. They said, wow, this is exactly what we are predicting with our models. So that was very, very interesting. And in hindsight, it somewhat makes sense that originally there were very few Lessepsian bioinvaders before the Aswan Dam was built, because the fresh water was a major barrier to that, and as soon as the Aswan Dam was built, then the gates were opened for biological invasion to happen. What are the future plans? So we are working more on looking at this drift selection differences. We are sequencing the entire genome of Pistellaria to make more sense of all the different results and know exactly which genomic regions are involved in selection. And I'm really interested in working with modelers in trying to predict what the future is of those guys, and it's possible to model exactly how biological invasions are going to happen in the Mediterranean. The Suez Canal is going to double in size very, very soon. That's what the plan is. So there is a paper that came out just a few weeks ago, double trouble, and there are huge problems with this. As you can imagine, all what I talked about is very bad, and by doubling the size of the Suez Canal, it's going to be 10 times worse, of course. The place is going to be a real disaster. The rate of biological invasion is exponentially increasing. Now it's going up, and I mean, I used to be able to keep up with the literature about what were the new biological invaders in the Mediterranean, and I almost lost track because in the old days, it was, I mean, the old days just 5 or 6 years ago, it would be 1 or 2 species a year. Now it's 10, 15, 20 species a year. It's really incredible how quickly it has happened. This is a picture that was taken in Italy. It's really great, with the difference that these guys are parrotfishes from the Mediterranean, but they are supposed to be in the south, and now they've moved northward because of warming ocean, and these are rabbit fishes that are not supposed to be there in the first place. Jack Briggs says that this is no big deal because the Mediterranean was originally the Tethyan Sea, and now it's just the same. So this is what a picture taken from Indonesia, which is the old Tethys, and now the Mediterranean is becoming exactly the same thing. So these are my various collaborators. Danny Golani and Ernesto Duro are the main collaborators. This is actually Ernesto reading Danny's book, pretending that he's reading, but as soon as the cops are not looking at us, he's jumping in the water and catching fish. Thank you very much. On recruits, fish recruits, and recruits cannot smell them or are not, they don't get the right cues to avoid them. And so where they are present, they tend to hit specifically recruits, which I think is the main problem with lionfish in the Caribbean. With ocean acidification, olfaction is going to go down in reef fishes, and so they won't, smaller fish won't be able to smell predators as well. That's one of the various issues. The big difference between lionfish and cornet fish, though, is that lionfish, I think, can be found in the Caribbean at very high densities, while cornet fish, while people notice them quite a bit, they have not yet been found in very high densities. So you see them, you go in the water, you see them one here or there, but you never have, I mean, I've seen pictures in the Caribbean of, hundreds of lionfish everywhere. You never have that in the Mediterranean. I don't know if it's because it's not there yet, I'm not sure. But in general, they are not super abundant. But that's what they do most of the time.
So since it seems like osmoregulation regulation seems to be important, has anyone looked to see whether the species that have invaded are more tolerant of low salinity conditions compared to, related species in the Red Sea that have not made it through?
So Yoni Bellmaker wrote a paper last year about, because the
the premise was that it was a stochastic event. You just grab any random fish in the Red Sea, and at some point, a lucky one just goes through. And instead, he was able to model to use a model where he could predict which species were the most likely candidates to go in. And now with their new model, they think that the ones that can make it are the ones that are able to breach all the various salinity barriers. That's what this paper is about. But these are predictive models, so they say this is what we think is going to happen based on the characteristics of the fish that have made it so far. So it's likely that this will be the case. I believe it, but we will see. We don't have to wait very long. Really, I mean, it's kind of interesting. Cass, what's up?
If you said the dam was built in '69,
Yeah.
why are some of these things happening now? You know, or is it just that it's
So,
studied?
so people have said that there really was a threshold, and the ocean has warmed gradually since then. And apparently, 5 years ago, that's when everything started happening, really. And so it was never, I think it was a combination of temperature and salinity. And once this happened, then things became, mind you, there were a number of migrants, but when I started this, which was not that long ago, I think that there were like 35 migrants, and now there are 90. And the stuff opened in 1869, so the rate of migration has increased incredibly. It's a big, big difference. Yeah.
I have a question about the bottlenecks and what your expectation would be under, how many generations would it take to where you would expect to see a bottleneck?
So Carlos Garza at NIMS has written several papers about this, where he modeled it exactly and has some algorithms to figure it out. But the rule of thumb is that it's roughly as many generations as there are individuals, roughly. So because there are relatively few individuals that go there, usually it's relatively few generations.
So if it's been 50 or 60 years and you're not seeing it,
Yeah, so then
you think the number is low, and with the increase in the size, you will expect more, and so you still would
that's the
expect it.
prediction. Well, that's. I think that when he was first interested in modeling that is when he worked on elephant seals, and in elephant seals, I think that the numbers were in the tens, if I remember well, in Guadalupe Island, and so that's how he figured out exactly how it was working.
Is there any way to enlarge the Suez Canal or to build a canal like they did with Panama, where they have steps that keep invaders from going through?
So the reason why Napoleon decided not to build a canal is because they made a mistake in their calculations, and they thought that the seawater level between the Red Sea and the Mediterranean was very different, and they said it's going to be too complicated. As it turns out, as opposed to the Pacific and the Caribbean,
Right.
the Red Sea and the Mediterranean are almost level. It's about a foot difference. So you cannot make those locks, exactly. But Bella Galil, who is the first author of this last paper that had lots of press called Double Trouble, she said, let's just make those locks in the bitter lakes, and they are not locks to allow you to change the level of the boat, but just to lock the boat for a long time and then wait for the water to become either high pH or change the water some way, kill everything, and then reopen the gates. This is a matter of money, of course.
Yeah.
So, it's very expensive
They
to
don't
do
want to
that.
stop.
No, not at all. And, I won't go into the details, but when the first author is Israeli and is criticizing the Egyptian government for not doing things very well, it's just difficult. It's really difficult. I mean, I wanted, the last paper I wrote it with this colleague from Lebanon, and the sort of the father of this entire field is from Israel, and I asked my Lebanese colleague if we could put in the acknowledgement some sort of an acknowledgement to his contributions, and he said, if I do that, I'll go to jail. I cannot do that.
Really?
Yeah, so he said, there's no way. He said, you can do it, but take my name out of the papers. Okay.
All right, well, let's thank Giacomo. He'll stick around.